one of our own, that having antibodies gives you a very high risk reduction in terms of reinfection, right? So it turns out that having antibodies is very protective. There are reinfections that can happen in rare cases, but infection usually provides better protection against infection, not just symptomatic infection, than vaccines. And so that data is now there, and I think it's safe to assume that we just don't know how much you have to have. But again, the next few months will probably bring that data.
Wonderful. And we're making reference, at least in general terms, to a variety of publications. I want to let our audience know that what I will do after our seminar tonight is to be in touch with Professor Kramer, and we'll put together a short list of maybe pertinent studies and overview reviews that we think might be of interest to you all. I would commend to you, if you're interested in vaccine development and all the different activities currently going on, a single author Nature review that Professor Kramer authored, published, I believe, in September of last year in Nature, essentially a wonderful overview of how vaccine development happens and the different candidates for the COVID vaccines. And there are a couple of follow-up publications that from Professor Kramer that I also want to list. Florian, I want to shift gears just briefly and have us talk a little bit about these virus variants, mutants in the UK and South Africa and South America. We're hearing about a variety of them. And just your sort of overall take, I saw an article in the New York Times yesterday that, you know, it's believed that in this country, here in the US, I believe the UK variant is believed to be doubling every 10 days and will soon be potentially the dominant type of virus in the US. Just at a high level, what should people know about these virus variants in terms of their infectiousness? And we've talked a little bit about the vaccine effectiveness against these mutants, but just remind us one more time, please.
Yeah, sure. And maybe I should start with how they might have emerged, right? They're very unusual because they have many mutations, and they exceed the mutation rate that we see for coronaviruses or SARS coronavirus too. So it's a little bit surprising that they showed up, and there's a hypothesis that they were actually generated in people who had more persistent infections and maybe a weak immune system, and there was kind of a constant low but still a selection pressure, and that made them evolve and A, better adapt to the human body, and B, maybe there was also some immune escape in those people involved. This is a hypothesis right now, but there are cases in cancer patients, for example, where there's persistent infection and where you see similar mutations show up. And so they were initially detected because they expanded, right? So the variant B117, which was first detected in the UK, was already detected in early fall at a very low frequency. And then it started to increase in frequency. And if you see that in the population, the assumption is that the virus variant has an advantage over other variants. That's why it expands. And so the B117 variant, which people call the British variant, but as a virologist, it's politically incorrect to name a country and the virus. That variant started to be dominant in the UK, and it is believed that it's approximately 35% more infectious, more transmissible. That doesn't sound a lot, but of course, you know, if you look at the numbers, large numbers and reproductive numbers, then of course it gets an advantage. That doesn't mean that these variants or this variant can be stopped by wearing a mask, by washing your hands, by socially distancing. Of course, those things work against viruses that are way more infectious, like measles, for example. So that's not really an issue, but they spread preferentially. And the same was true for the B1351 variant, which emerged in South Africa, was detected in the same way. And it's also true for the P1 variant, which emerged in Brazil. So these variants have a number of mutations. One at position 501, that's in the spike protein in the receptor binding domain, that seems to allow them to be more transmissible. And so that's what the British variant, that's the main mutation of the British variant. There is more. But what we know about the B117 variant from the UK is that, yes, it's more infectious. It doesn't cause more severe disease, even though Boris Johnson at some point mentioned it, but I don't think the data was there to draw that conclusion. And we know by now that the vaccines work relatively well against that variant. So there is not really a sharp drop in vaccine efficacy. The vaccines that we're getting vaccinated with should be fine against that virus. The South African B1351 variant and the Brazilian variant have additional mutations, and they're more problematic because they actually influence neutralizing activity. And it has been shown in vitro that sera from people vaccinated with the Moderna, the Pfizer vaccine, for example, have a reduction in neutralizing activity against these variants. The neutralizing activity is not gone, it's reduced. And the remaining activity is most likely enough to protect ourselves. And it has also been shown for the Novavax and the J&J vaccine, as I mentioned, that yes, the efficacy suffers a little bit, but it's certainly not gone. And you have to keep in mind that if the efficacy against mild disease goes down a little, then usually the efficacy against severe disease goes down even less. And so, of course, we need to keep an eye on these variants, but at least in the US, they are right now not a major issue. The issue that South Africa is facing right now is that the AstraZeneca vaccine does not seem to work against the variant in terms of mild to moderate disease. And there's not enough data to conclude that it still works against severe disease. And of course, if you don't have that data, you don't want to roll it out and vaccinate your population, and then just to find out that it doesn't work at all. That's unlikely, but that's why South Africa decided to stop the rollout. So that's where we stand with these variants. We'll see an expansion of the B117 variant in the US. I hope we'll see less of an expansion of the B1351, the South African variant, and the Brazilian variant. We'll see how that goes. If they expand, it's very likely that the vaccines will still give good protection. And if we figure out in the end that the effectiveness in the population is too low, vaccines can be changed to reflect that sequence.
And I was going to ask you about that last point, if I may. So assume, this is an RNA virus. There's a higher rate of mutations in RNA viruses. There may be certain situations, as you point out, where that might even be accelerated in patients with prolonged infections. So, and we're barely 12 months into the COVID-19 pandemic, and we're already seeing some of these mutants. So mutations? Is it likely that there will be more, I assume? Can we assume that some of these mutations will eventually be such that some or most vaccines may not work? And therefore, my question would be about vaccine development. You've published on this quite a bit, explaining how much vaccine development might be shortened to respond to new challenges, new viruses, new mutations. Talk to us a little bit about that, please.
Yeah, I mean, it's obviously getting easier right now because for the RNA vaccines, you can basically change the sequence overnight and produce a vaccine that now is matched to new variants, right? Or to one of the new variants. that's complicated a little bit about because of course you want to have the data that in humans that new vaccine now does the same that the old vaccine did against the old variant. So you probably need a small clinical trial with neutralizing activity, immune responses as readout. I don't think you need a phase 3 trial, but that takes a little bit of time. And the other complication, of course, is you don't know if you have the sequence now of the new variant in your vaccine, if that vaccine still protects against the old variants or the old virus, which might still be around, right? So it's getting a little bit complicated there, and companies are working out pretty good concepts to get around that. Moderna was proposing or testing to give a third shot, but now with the modified vaccine as third dose, Novavax is talking about making a bivalent vaccine. So there's ways to do that. And I think if need be, the FDA will also be relatively lenient because there's already a lot of safety data with the original sequence and the platforms, right? So I think that could be done very quickly. But in general, with these mutations, if you would have asked me 6 months ago, I would have told you I wouldn't worry about the stability of the virus because coronaviruses, in contrast to other RNA viruses, have a proofreading activity, and they actually move relatively slowly on an evolutionary trajectory if you look at other human coronaviruses. And if you compare it with influenza, what you typically see with flu, when a new pandemic hits, there is initially a quicker evolution because the virus still adapts to humans, and then it doesn't move that much for 2 or 3 years, and then antigenic drift starts. So in a way, what we see now is even faster than influenza, but it might just be that initial adaptation that influenza does as well, and it might not be bona fide antigenic drift. So it's going to be very interesting to see how this develops. It could be that the virus keeps changing, but it could also be that it gets stuck and stays where it is in its evolutionary trajectory. And I'm still hoping for that. So we'll see. But technologically, I think we have solutions if we need to find them.
And it sounds like maybe at some point it could be as simple as having a cocktail of mRNAs injected. Can some of the concepts that you have been working on for flu vaccines in terms of a universal vaccine eventually apply to, say, a universal SARS-CoV-2 vaccine? Will we, I know that is speculation today, but down the road, 10 years and more down the road, will there be a universal vaccine, do you think, against SARS-CoV-2 covering essentially all variants?
Yeah, that's an exciting topic, and a lot of people are thinking along those lines right now. It might be much broader than SARS-CoV-2, right? SARS-CoV-2 is a beta coronavirus, like 2 of the human coronaviruses that are circulating, like the old SARS virus, like MERS, the Middle Eastern Respiratory Syndrome virus. And so it would be nice to have a vaccine that protects against all of those. And I think similar approaches that compared to what we are using for influenza, could work here as well. It's just you can't develop that in a year. It takes much more time. Not as much time as it took initially for influenza to get us started, just because the technology is better. But still, it's not going to be an overnight development. But that would be ideal because there might be another coronavirus pandemic at some point, and we want to be prepared for that too, I guess.
Well, it's a good thing. It takes a while. That's going to keep you busy for a while, Florian. That's always good. Let me ask you, and this is getting just slightly away from sort of core vaccine questions, but Christian Drosten, who's a virologist in Germany, who, as you well know, has been a fairly prominent voice in that country in recent months, is, I believe, one of the co-discoverers of the first SARS-CoV virus. He has opined that eventually down the road, he sees SARS-CoV-2 or COVID-19 infection sort of take its place, almost among the other human coronaviruses, once there's sufficient immunity worldwide, there's widespread vaccination, there's widespread herd immunity, whatever you want to call it, that the virus may or may not go away completely, but reinfections might essentially be somewhat more like a common cold. Is that a scenario that seems likely, or can we predict where COVID-19 will eventually be 10 years from now as a disease, as an infection?
No, I mean, I think that's exactly where it's going to go. We will not eradicate this virus. We haven't even eradicated polio yet, and polio should be easier than a respiratory virus. So I think I don't think that that is a realistic option. And I think he's right. I think once people, even if they're protected, if their immunity goes down a little bit, they probably can get reinfected asymptomatically or mildly symptomatically, which might then lead to another boost in immunity, and then you're good for a few years again. And I don't think that the virus will go away necessarily, but the impact will be relatively low. Because a lot of people will have pre-existing immunity and will be protected from any kind of moderate or severe disease. And B, once a lot of people have at least partial immunity, the circulation of the virus is going down. I mean, we're talking about herd immunity, and that's exactly what that is. We already assume that with vaccination, the circulation will go down. And once there is fewer viruses circulating, or there's little virus circulating, the chance of getting infected is low. And if you have pre-existing immunity, even if you get infected, there's not really consequences, right? And so that will basically remove the impact on society that we see right now. But yeah, I don't think it's going away. I think it's going to become the fifth human coronavirus.
Yeah, interesting. I want to be mindful of your time. I have, if I may, one last member question that was submitted just yesterday to me, and then we'll wrap it up. That question relates to folks who've been known to have been infected with COVID-19 and have recovered. Should they also be vaccinated? And if so, do they need the same vaccination regimen? And I know you have actually researched and recently published on this. And I was wondering if you could tell us what your thoughts are. Currently, my understanding is there's no distinction being made in vaccination programs as to how to handle folks who've had COVID-19. Where do you think that issue is going?
Yeah, you're right. The recommendation right now is, even if you had already COVID-19, you're supposed to get 2 doses of vaccine. And it makes sense to still get vaccinated even though you already have antibodies, because what we see in natural infection, the antibody levels are variable, right? Some people have high titers, some people have low titers. After vaccination, that becomes very uniformly high. So that's what we see when people get vaccinated. Everybody has high titers. It's in the top 25 of what you see after natural infection. Now, the question is, should you get vaccinated twice? And that's a different story. Immunologically, if you haven't had COVID and you get 2 doses, you get a prime and a booster dose. And that's beneficial. That gives you high antibody titers. It drives affinity maturation. It probably is also good for longevity of your immune response. Now, if somebody already had COVID, that person is already primed. And if that person now gets the first vaccination, that's the booster dose. And so our question was, if you look now at antibody titers and how robust they are, is that really the case? And we looked at that, and it is the case. People who had COVID and then get their first shot have antibody responses that are equal or better as compared to people who didn't have COVID and then got the second shot. And so we had this finding a few weeks ago, and in the meantime, there's now a total of 4 papers, all in the preprint stage, but pretty good groups and solid data that, and all of them show the same. And it seems from one of the papers that people who had COVID and they then get the vaccine have a very, very good neutralizing response also against these variants, including the South African variant. And the other point that that came up is that a lot of people, when they get their second shot of RNA vaccine, have stronger side effects than after they get the first shot, right? Usually the local side effects, the injection site pain is the same, but the second time systemic responses like chills, maybe elevated temperature, fatigue, muscle pain, etc., it's usually occurring at a higher rate. Now, we have initially heard, just anecdotally, that people who had COVID and they got their first shot have strong side effects that were comparable to what other people got after their second dose. And so we looked at that too, and we quantified that, and it's actually the case. So right now, from a research perspective, and I can't say this from a regulatory or policy perspective, it makes sense to give 1 dose to somebody who already had COVID. The second dose might not be necessary at all. And so right now we are trying to actually stimulate a discussion to change the policy. That might or might not happen, but again, there was a lot of data that was produced in the last few weeks, and independently of where the data was produced, the results are the same. And so I think we have a pretty strong case. But until the policy changes, I would recommend to go with the 2 doses that the CDC is recommending.
Yeah, very good and understood. And I very much appreciate your time tonight. There's clearly lots and lots of things that are still to be learned about the virus itself, the vaccination efficacy, the different approaches to different variants, and lots of other questions. But the last hour, I hope for our audience, for me certainly was extremely informative, and I very, very much appreciate your time. I can't let you go before asking you about your tweet from a few days ago where you showed your skis and your commute to work through the New York snow. I hope you're not sitting there ready to put on your skis to go home. Are you walking again, or are you truly skiing to work these days?
So usually if it's snowing in New York and there's enough snow in Central Park, I take the opportunity to use my cross-country skis to go to work. And I did that actually twice, but today I'm walking. But it's a lot of fun, and I'm originally from Austria. I like snow, I like skiing, and I don't get to ski too often. So you have to take these opportunities and actually do it.
I was going to say, after that tweet, I was starting to think about the little hills I remember in Central Park, and they don't compare to Austria, that's for sure. Listen, Florian, I very, very much appreciate your time and all the information. It's been tremendously helpful. Thank you a lot.
Thanks for having me.
Thank you, Dr. Margaret and Dr. Kramer, for a great discussion tonight. And thanks to all of you who participated in this evening's webinar. As a participant tonight, ASGE will circulate a short survey in the next 24 hours. Your feedback would be greatly appreciated and helps us growing our webinar topics and presentations. Finally, don't forget that you can access the recordings of this webinar and engage your peers by accessing the archives on GI Leap at learn.asge.org. This concludes our presentation. We hope tonight's information is useful to you in your practice.